go ahead and get started now that we're 3 minutes past. I think that's kind of the lucky number here to make sure that everybody's in. So I wanted to start just with a couple of logistical things. First, welcome to our second Distinguished Ecologist lecture series, seminar series. Please make sure, I had this set to auto mute, but just in case, please make sure you do mute your audio. You're welcome to keep your video on if you're comfortable with that. We'll hopefully have time for questions after the talk, and I'll go through the logistics of that then, but the short of it is just use the raise hand button under the reactions menu at probably the bottom of your Zoom window. And also please save any questions that you might want to post in the chat until after the talk. Sometimes that can be distracting during the talk. And then finally, if you are enrolled in Ecol 571, don't forget that tomorrow you guys have your lunchtime meeting with Jonathan on Zoom, and so we'll do that then. I am very pleased to introduce Jonathan Losos. I've known Jonathan for a long time now, and I'm truly honored to have him here as one of GDPE's distinguished ecologists. Jonathan is perhaps best known for his work on Caribbean Anoles lizards, and more specifically on how similar ecological niches drive repeated evolution of both morphology and behavior across different Caribbean islands. Jonathan got his start as an ecologist early on. I'll focus just on what happened when he started at Harvard, because there's a lot even before that. But while completing his bachelor's at Harvard, he started his formal work with Anoles, which resulted in 2 publications as an undergrad. And then from there, he moved on into Harry Green's lab at Berkeley, who many of you know from his sabbatical here just a short while ago. Thank you, Chris. And Jonathan completed his PhD on how Anoles species adapted to different environments. And then the rest, as they say, is history. So Jonathan has authored over 240 peer-reviewed publications, as well as contributed to over 30 reviews, commentaries, white papers, and popular articles. He has co-authored textbooks on biology and the living world, and one of which we actually use for our Life 102, 103 series here at CSU. And he has written 7 books, including his most recent, Improbable Destinies, which many of our honors students have read, as well as several of our graduate TAs for BZ 220. Jonathan has always had a very deep dedication to undergraduate and graduate education. He has a long list of academic descendants from his time as faculty member at Washington University in St. Louis, and then at Harvard, and now back at WashU. He's currently the William H. Danforth Distinguished University Professor at WashU, and he is also the director of the Living Earth Collaborative, which is a partnership between WashU, the Missouri Botanical Gardens, and the St. Louis Zoo, with an effort to advance knowledge of biodiversity and ensure the future of Earth's species. So please join me in welcoming Dr. Jonathan Losos for his talk on using experiments in nature to study evolution in real time, research on lizard adaptation in the Bahamas.
Well, thank you very much, Jennifer, for that, that very nice introduction. And Jennifer didn't mention that I've known her since she was a graduate student at Washington University, and maybe tomorrow with the graduate students, I can tell some stories from those times. Just a little threat for Jennifer. But anyway, it's a real pleasure to be here. And of course, I wish I really were there instead of where I am, but oh well, that's the way the world is these days. And so I'm certainly honored to be chosen to be part of this wonderful series that you guys have. So with that, let me get going. My talk is going to be, in some respects, a review of my career. And I hope you'll indulge me, but the research that I've been doing on the Bahamas has a timeline that goes back to the very beginning. And so this talk is an opportunity for me kind of in some ways to reflect how I've gotten to where I am today. So I hope you don't mind the historical perspective. I hope perhaps it's a little useful for the graduate students out there to kind of see how a career unwinds and goes in ways one never would have expected. Well, having said that, let me begin. We'll begin when I was much younger than I am now. These are pictures of me as an undergraduate and a graduate student. And I want to start with a talk I heard in a series very much like this when I was an undergraduate, a junior in college. The weekly biology department seminar featured John Endler, who many of you probably know, giving a talk on his research, his contemporary research at that time on natural selection on this fish here, which you probably all recognize as a guppy. What many people don't know, however, is where guppies actually come from. They actually do look kind of like that in the wild. This is a guppy from its native habitat in Trinidad. Guppies live in mountainous streams in Trinidad and Venezuela. And one thing that John Endler took as his starting point was the observation not by him, but by people before him, that guppies were not colorful everywhere. In some places, they looked like that on the screen, but in other places, they were quite bland. And the question that he started out asking is, why do you see this variation in color? And I should point out, this is mostly the males that are very colorful. Females aren't very colorful anywhere. Well, there was an association that existed between the presence of larger predatory fish, such as this pike cichlid, and the lack of color in the guppies, that when you had predators, the guppies were not very colorful. And so the idea, which obviously suggests itself, is that in the presence of a predator, a guppy could not afford to be colorful. It would be too obvious, it would get eaten. Whereas in the absence, for some reason, being colorful was beneficial to males, presumably because females prefer colorful males, something that we now know to be the case. Why that is another story. Well, that was the association that Endler started with, but as we all know, correlation does not prove causation. And so Endler decided to do an experiment. And so what he did was he basically airlifted some drab guppies, not very far, just slightly up a stream, to a pool where there were no guppies and no predators. And the prediction was, in the absence of predators, they should evolve to become more colorful. And indeed, that's exactly what happened over a remarkably short period of time, just a couple of years, a few guppy generations. He then repeated that experiment in the laboratory, where he created artificial but very realistic pools with and without predators. And as you can see, over a period of 14 months, populations diverged in the same way, becoming much more colorful in the absence of predation. Well, this talk was seen as spectacular by everyone in the audience. I remember walking out and listening to the graduate students whom I hung around with, basically saying, who knew that ecology could be an experimental science? And it was really a stunning demonstration that not only does evolution, can it occur quickly, but you can actually do experiments, use the gold standard of science to test evolutionary hypotheses. All right, so that I saw that talk in my junior year in college. It made a big impression on me. I mean, I can still remember it to this day and tell you about it. A few years later, while I was a graduate student in Berkeley doing comparative studies on Anoles lizards, I came across this paper by Tom and Amy Shaner, where they had done experiments in the Bahamas. And what they did was that they found that on small islands in the Bahamas, there were no lizards. Everywhere else, there were Anoles lizards, except on the tiniest islands. And so they had the idea, let's put lizards on these tiny islands and watch them go extinct to understand something about the extinction process, something we still need to know about today. And so what they did, this is, I should say, our focal lizard for most of this talk, the brown anole, Anoles sagrii, the workhorse of Anoles biology. What they did is they put Anoles lizards on tiny little islands, expecting them to go extinct. Well, much to their surprise, on all but the smallest islands, the populations thrived and they did perfectly well. You can see the axis here on islands, basically a little more than 10 square meters in area. That's a pretty small area. On any island larger than that, you put lizards there and they will survive. On the smaller islands, not so much. Well, that led me to a thought. So let me put it in another way. What Shaner and Shaner had done is they had created lizard populations on about 14 islands, and those populations had been running for 15 years. Now, I surmised, all right, I'm sorry, I can't see my next slide, so I'll be a little guessing here. My PhD work had been at the comparative level, and a major part of it had been looking at evolution of limb length throughout anoles. And and there had been an association between the breadth of a surface a lizard used and how long their legs were. And multiple times species had evolved short legs to use narrow surfaces. So on short legs, anoles have very short legs. They move very carefully. On broad surfaces, they have long legs. We've seen this phylogenetically. And the explanation is that to move on a narrow surface, you need to be able to move very carefully so you don't topple off. Whereas on a broad surface, longer legs let you run faster. But again, this is a correlation. And so remembering Endler's work, I thought to myself, well, Shaner has essentially done an experiment to test whether vegetational differences drive lizard limb evolution, because I surmised that the islands differed in their vegetation. So I approached Tom Shaner and basically asked him, have you ever thought, has it occurred to you that you've essentially done an evolution experiment? Have you thought of going back to see if the species have adapted to differences among the islands? And his response was what I could only have hoped for in my wildest dreams, but never really expected. He responded, that's a great idea. Why don't you come do a postdoc and look into it? You know, I never really expected to hear that. So there's a lesson in this. It never hurts to ask or to point things out. So in any case, I went to UC Davis as a postdoc and started the work I'm going to describe to you today. And that work has continued, as you'll see, in a kind of linear progression for almost 30 years now, 30 years plus now. So here's the roadmap for the talk. It's, as I say, a play in 4 acts. First, we'll look to adaptation to different environments, what I was just telling you about. Then the role of interspecific competition in driving evolution. Then the role of predation. Then we'll have a short segment on founder effects. And then if I have time, there'll be an encore that I'm sure you will demand. So let's get started. I have to tell you the cast of this talk. All this work has been done with Tom Shaner, my postdoctoral mentor and now a friend and colleague for many years. Dave Spiller, a long-term postdoc, long-term research associate with Tom. And then more recently with 2 people who got PhDs in my lab and are now faculty, now full professors in their own right, Manuel Leal at the University of Missouri and Jason Colby at the University of Rhode Island. All right, let's get started. Act 1, adaptation to different environments. This is work going on in the central Bahamas, in this case, on an island called Staniel Key. And on Staniel Key, there are many small islands, such as these here. And let me show you the islands we're working on this map. We're looking at these ones here. Here is the legend, so that's a kilometer. So these are small islands. These are tiny islands, maybe the size of a baseball diamond. So these are very small islands, and these are the islands that Tom Shaner and Amy Shaner put lizards on to see what would happen. And just to show you that my surmisal was correct, they did differ in vegetation. This is one of the more vegetated islands with a number of small trees. On the other hand, some of the islands were very scrubby with only very narrow vegetation. So there was variation in vegetation. As I said, the prediction was that the lizards over the course of 15 years or so would have evolved differences, that the lizards on islands on which the vegetation was narrower would have evolved shorter legs. So, and here's just a picture of a brown anole on different surfaces. So this is me. I love this picture from early days in the work. Ignore my socks, please, and just look at what I'm doing. We would go to these islands, we would catch the lizards, and we would actually measure them right then and there. What I'm looking through is something called an ocular micrometer, where I'm counting the number of scales on their toes. In addition, I would take a little ruler and measure how long the lizard's legs were. I'd collect all the data and then just put the lizard right back where it was caught. And so I went back to the islands that the Shaners had established, the populations at this point, it was 15 years earlier, and measured their limb length. And here's what we found, exactly as we predicted. I reversed the axes from what should have been the logical way. It should have been perch diameter on the x-axis. But in any case, each dot is the mean value of the lizards on an island, the diameter of the surface they used, and how long their hind limbs were relative to body size. And as you'll see, there was a pretty strong statistically significant relationship. And what this indicated was over the course of really 15 years, which is about 1 to 2 lizard generations, there had been adaptive divergence to different environments. So this was, I should point out, in the early 1990s. And so we submitted the paper and it ended up getting published in Nature. And as you may or may not know, when Nature, every week they come out with a new issue, they release a press release where they choose a few papers to hype as, to try to get press interest. And they, for whatever reason, chose this paper. And so I still have the press release they sent out, and here's what it said. This may be among the most important work in evolutionary studies since Darwin studied the diversity of finches on the Galapagos Islands during the voyage of the Beagle. Well, it was a nice study, but this was ridiculous. That's just way over the top. But anyway, they sent this press release out to media all around the world. Well, we had just left for the Bahamas for our yearly field trip. And at the very last moment, remember, this is the mid-1990s when there was no internet in the Bahamas. And in fact, we did not have a phone in our room where we were staying. But at the very last minute, I put on my voicemail, if you need to reach me, call the hotel, the manager of the place we're staying. Well, here's the place we were staying, the Lofty Fig Villas. And right here, I hope you can see my pointer, this little pink shack is where the manager would stay every day. And one day we come back from the field all hot and sweaty, and he calls me over and he says, the New York Times wants to talk to you. And so the next day I call up the New York Times, and I come back the next night. It's Boston Globe and USA Today have called. And for a couple of days, ABC News called. They wanted to send a film crew. The manager of the place was really quite annoyed because I was tying up his only phone, and he couldn't believe these knuckleheads. He thought we were, these people went out and caught lizards. He didn't know what was going on. Anyway, we got all this publicity, and it was on the front page of the Boston Globe. It was prominently displayed in the New York Times and so on. I can tell you part of the reason that where I am today is this publicity we got, all because of Nature went over the top with their press release. So it just shows you how the contingencies of academic careers. So that's how this work first got published and people got to know of it. However, I would go around and I would give talks. And always when I'd give talks like I'm giving today, there would be someone who would raise their hand with a pesky question. And it was always one of those damn botanists. And they basically asked, what about phenotypic plasticity? Could it be that the short legs of a lizard are just because it grows up using narrow surfaces? Just like you water a plant, it grows bigger. Maybe you put a lizard on a broad surface, it grows longer legs. Well, this seemed like a ridiculous idea to me, but I kept on getting the question. And so I figured I need to look into the literature to see what we know about limb development in lizards. So I went to the library, and that turned out to be very easy because there was absolutely no literature at all on environmental effects on limb length in lizards. However, I'm being serenaded by my cat. However, there was a literature that was pertinent. And I must say that this is among the most bizarre literatures I have ever read in my life. I once gave this talk and said that, and there was someone who does that sort of work in the audience. So sorry if you do this sort of work, but it was people who were looking at the effect of exercise on vertebrate limb development. And so they did things like this. They would take a group of mice, and as they were growing them up, they would make them run on a wheel for 10 hours every day. And then they would have a control group that wouldn't get a wheel, so they didn't get to run around. And the goal was to see how does exercise affect the developing limb? Or they put a rat in a bathtub and made it tread water for, I think, 4 hours a day. And again, the control group, they didn't have to do that. Or they took a chicken and they put it on a treadmill and had it walk on the treadmill for a long time. Now you're thinking to yourself, that's not a chicken. I know it's not a chicken, but it turns out if you Google chicken treadmill, you don't get any useful figures. So I used this instead. Although once I gave this talk a little while ago, and immediately when it was over, somebody sent me this as a better indication. So maybe that works. Anyway, there are all these crazy studies where they made chickens and mice and so on do these things to look at the effect of exercise on development. And what they found was, in fact, that as a general rule, if you make animals exercise more, they develop thicker bones. And that's because it turns out that bone is a very labile substance. I didn't know that. And you put more stress on a bone, more bone will be deposited to make the bone stronger. That's why weightlifters have very thick arms. However, the finding we had was not the thickness of the bone, but the length of the bone. And there was almost no evidence that exercise affected the length of the bone, with one notable exception. There was an old study from the 1950s on professional tennis players, male professional tennis players. And what they did was they measured the length of the serving arm of male of pro tennis players. Now, if you think about it, somebody who's a pro tennis player throughout his formative growing years has been smacking balls for hours every day of his life. And so the question is, does that affect the length of the arm of the adult pro tennis player? And conveniently, there's actually a control for this. Their non-serving arm. And so what they did was they measured the length of the serving arm and the non-serving arm. And sure enough, pro tennis players have longer serving arms. And I should say this was measured with X-ray. So it wasn't the arm was hanging more loosely from the ligaments or whatever. The bone itself was longer. Now, you might be thinking to yourself, maybe the causality is reversed. Maybe it's asymmetric armed people who become tennis players. That could be the case. But if we throw that aside, it does suggest that a lot of putting stresses and strains on a bone actually can cause it to grow longer. So there was only one thing to do, and that was to do an experiment. And so to do this, at the St. Louis Zoo, we got a bunch of baby brown anoles, and we raised them in two conditions. One was on a broad 2x4, like this lizard here. Now, this is a typical habitat for a brown anoles. They sit on broad surfaces, and that's how they sit. And so this is a happy anole. On the other hand, this is the other treatment. This is not a happy anole. We made him hunker down on a narrow dowel. So we grew the lizards in these two conditions until they were more or less adult. And then we looked at their hind limbs. And here's what we found. First, we found that males had longer hind limbs than females once size is removed, size effects. This is not news. We knew that this was a sexually dimorphic trait. But we compared the narrow and the broad surfaces, and sure enough, for both males and females, lizards raised on broad surfaces had somewhat longer legs than lizards raised on narrow surfaces. That it is a phenotypically plastic trait. We were so surprised at this finding that we did the experiment a second time and got the same result. Several other people have redone the experiment and for the most part gotten the same result as well. So there is phenotypic plasticity in limb length affected by the posture that lizards adopt when you make them use different surfaces. Now, the obvious question, well, if this is a genetically based trait, what are the genes? And we're getting there. It's for years I've been getting that question. I'm saying someday we'll find out what the genes are for limb length and we'll be able to actually test directly for a genetic difference. We're not there yet, but we're getting pretty close. And I think in the next couple of years, we'll have a chance of finding such genes. There's a lot of very cool work on Anoles genomics going on. I will point out though, that the amount of difference produced in this plasticity experiment, this is a red bar shows the difference between the two treatments, is this much. And compare that to our experimental populations, our experiments generated much greater difference in limb length than we could get in this experiment. And this experiment was a pretty extreme difference for lizards. I mean, we made them use differences about something like this. And so this strongly suggests that although plasticity may be a component of what we found, probably the greatest effect was genetic differentiation. Well, with that, I will now go to the second part of the talk, which has to do with interspecific competition. And so once I got my faculty position at WashU, I decided I wanted to do my own evolution experiment. And so we moved from slightly to the south in the middle of the Bahamas and the Exumas to a town, an island called Great Exuma and a town named Georgetown. And there we looked at the interactions between two anoles, the brown anole and the second species, the green anole, Anoles smaragdinus. Now, if you know the Carolina anole, Anoles carolinensis in the southeastern United States, the green anole is a very close relative, a very similar species. So the green anole is more arboreal than the brown anole where they co-occur. So we took advantage of, again, a bunch of very small islands that did not have any lizards. And we found 16 islands and we broke them into 4 treatments. We had one treatment that got no lizards because we were also looking at the ecological effects of having lizards on the islands. And then the other 3 treatments, some islands got 5 brown anoles, some got 5 green anoles, and some got 5 of both. And it was 2 males and 3 females. And so the question is, does the presence of the other species affect each of our species? So we had 3 lizard treatments. Each one of them replicated 5 times. I told you the wrong number of islands. We had 20 islands, 5 per treatment. So 3 questions. Can brown anoles survive on islands smaller than those upon which you normally find them? Can green anoles survive on islands? and how do they interact with each other. So we'll start first with the brown anoles. Here's year one. So we introduced 5 lizards. A year later, they've all at least doubled in population size, and one of them has really exploded. 3 years later, 9 of the populations are very well established. For the green anoles, it was a different story. Although they do okay for the most part in the first year, by 3 years in, 4 populations have gone extinct. 2 other populations are down to a single lizard, and that lizard is a male. So that doesn't look so good for those either. So something seems to be bad about these islands for green anoles. We suspect it's some combination of heat and humidity, but we've never actually figured that out. We can understand as a result why it is. Well, all right, what about where we put the 2 species together? What we find, so there are 5 blocks of 3 islands. So each block had a green anole island, a brown anole island, and both. And what you see shown here is the density of green anoles. The red line is the green anole by itself. The yellow line is the green anole in the presence of brown anoles. And what you find is that in the middle of the experiment, when things are good, in all 5 cases, the red line is higher than the yellow line, indicating that green anole densities are higher when they occur by themselves, that brown anoles have a negative effect on green anoles. So there is an interspecific interaction between the species. So I think we can understand why green anoles aren't found on these small islands. Number one, some of the conditions simply are unsuitable. And then if you get brown anoles, that's even worse. But why don't we find brown anoles on these very small islands? You put them there, they do just fine. Something must come along every now and then that keeps the brown anoles from being there. Now, when you say something must come along every now and then, and you're in the Bahamas, obviously you're talking about hurricanes. And so in fact, when Tom Shaner published his first paper, Tom and Amy, on these islands, they said that it must be hurricanes. But the Shaners had the bad luck of starting to do their research in the Bahamas just after a natural lull in hurricane activity had started in the early 1970s. And so even though Shaner by this point had been working in the Bahamas for a quarter of a century, he had never seen the effect of a hurricane. However, in 1996, as we finished the 3rd year of our monitoring of this experiment, Hurricane Lily was churning its way over Cuba. And we had mixed feelings about this because on the one hand, we were dying, I should say it was headed straight towards our islands. And on the one hand, we were really excited to find out what effect a hurricane might have. But on the other hand, as I just indicated, we were still on the islands. And so we weren't so thrilled. So this is a map that was given to us. The bank in the Bahamas handed out maps, and you could track the hurricane because every 3 hours, the BBC, the Bahamas Broadcast Corporation, would give the coordinates of the storm. And so you could map the storm. And what we could immediately see is that as soon as the hurricane passed over Cuba, it took a hard turn to the right. And so it was no longer headed towards us. And then it turned right back, and it was coming closer and closer. And we can see it was heading right towards us. And in fact, the eye of the hurricane went straight over us. Well, the effects of the hurricane were pretty dramatic. We went out the next day. There were trees toppled all over the place. We counted 17 yachts that had been destroyed, either sunk or beached. And it was a big deal. But once we cleaned up, 2 days later, we went back out to see what our islands were like. And I should mention that just by good fortune, we had just finished our census for the year. So we had censused all the islands in the previous 2 weeks. And so we could now come back and say what effect the hurricane had, with a very narrow time window. It was like this a week ago, now it's like this. So we got our motorboat out of the tree it ended up in, and somehow I failed to get a picture of that, put it back in the water and went back to look at our islands. Now, one thing I didn't mention is that our experimental islands were situated on both sides of Great Exuma, some in the southwest, then the islands, some in the northeast. So we first went to the islands in the northeast. And what we found was that the lizard populations were down by 1/3 to 40%. I should say as well, the islands, you go on these islands, you could tell something had happened. Branches were broken, leaves were stripped off. Clearly, there had been some event, but by the same token, it was a quantitative effect. The islands and the populations had been damaged. We then went to the other islands on the southwest side of Great Exuma. This was my favorite island in the study. This is the one island that the green anoles were thriving on. They were like little grasshoppers in these bushes. You'd walk through the island and the green anoles would just go flying left and right out of your way. This was the island after the storm. We couldn't recognize it. We thought we were in the wrong place. All of the bushes essentially were gone, except for this stuff growing out of the limestone. This is another island. This had been a big 10-foot tall tree with a big drooping canopy, just snapped in half. And you can see everything else has gone on the island. And here's another island as well. So the islands were just trashed by the hurricane. We went to census the lizard populations. It turned out that was pretty easy. There were no lizards on the islands. So that was the answer to the question, why are these islands empty? As Shaner had predicted, periodically a hurricane comes along, clears the lizards off the islands. But the last question, of course, is why did the lizards survive on one set of islands and not on the other? Now, I should point out that it is known that hurricanes slow down. So the hurricane came from the southwest, hit the islands where the lizards were all gone, then went over Great Exuma, then hit the other islands. Hurricanes slow down when they go over land, but Great Exuma is only 1 mile wide, and that's not enough land to significantly slow down the hurricane. So it wasn't that the wind was less swirly on the other side. There was, however, one clue what had happened on some of our islands in the southwest. And here it is. This was on top of the island. And this is, of course, a starfish. Though I don't know if you teach marine biology at Colorado State, but people who know marine biology know that starfish do not live on the top of islands. And so obviously, what we all now know happened because of all the hurricanes in the last few years, the island was underwater. There was a storm surge of about 5 meters. These islands are at most 3 meters above sea level. So this storm surge hit the islands with great force, probably stripping off all the vegetation, and then submerged the islands for about 6 hours. And so between the stripping and the drowning, all the lizards were gone. So that solves the mystery of why these islands are empty. It also ended our experiment, but that was okay. I was kind of tired of that experiment. We could see where it was heading ecologically. It gave us an excuse to start a new experiment, and that is now part 3, predation. As you well know, over the last several decades, people have become very interested in the role of predation in regulating community structure. And in particular, one aspect, we were interested in the role of predation and what it might show. So we now move to the northern part of the Bahamas, to the island of Abaco in the town of Marsh Harbor, where I'll be staying for most of the rest of this talk. And I need to introduce a new player, and that is this lizard here, the curly-tailed lizard. So named for obvious reasons. If you as a predator or a potential predator approach the lizard, it curls its tail, essentially telling you, I see you, don't waste your time trying to chase me. But that's not why we were interested in it. We were interested because curly tails are substantially larger than anoles. And in fact, they are known to eat anoles. And the question is, what effect would the curly tail have on the habitat use and potentially the adaptation of brown anoles? Now, I must say this is an experiment that certainly of all the experiments I've ever done, this is the one we were the most uncertain of what the outcome might be. We knew that curly tails would eat brown anoles, but we didn't know whether this was just a rare once in a blue moon event with no greater significance, or whether they would have a major impact on the brown anole populations. So what we did is we found 12 islands. This time, these were islands that already had brown anole populations on them. And we randomly selected 6 of the islands to receive curly tail populations, with the other 6 as controls. I had the good fortune of working with the Howard Hughes Medical Institute to make a film about this research, and they explain it much better than I can. So let me show you a little snippet from this film. Let's see, here we go. If I get this right, share this again. Oh, that didn't work. I always blow this. Click on this. There we go. Can you see that? Okay, here we go.
Jonathan wondered if he could also create an experiment that would lead natural selection to favor longer legs.
I have to interrupt. I forgot to say, I had a huge argument with HHMI because this was not my project. It was in collaboration with all the people I mentioned earlier, and they refused to list them all. But this is a collaborative project between me, Shaner, Spiller, Leal, and Colby. some of whom are right here.
He found islands that already had anoles that lived primarily on the ground. His team caught and measured each lizard.
That's Jason Colby.
Then he brought in a predator, a species of larger curly-tailed lizards that eat anoles.
Here's the bad boy of our experiment. This is the curly-tailed lizard. Now, what we did is we added them to half of our islands. These are very voracious predators. What we wanted to know was, what effect would this guy have on the anoles on the island?
Would the anoles evolve longer legs to run faster, or larger body size to defend themselves?
Well, excellent question. Let me get back to my PowerPoint. Oh, I blew it again. Where'd it go? There it is. Okay, so here's what happened. You put the curly tails on the island and immediately you see a drop in population size. Here's where we start. The black dots have the curly tails and instantly the population is cut in half. And what is happening here, the population is going up in the control islands because the babies are being born. It's in the spring. And clearly the curly tails are eating a lot of the babies and probably some of the adults. And for the course of the experiment, there was a twofold difference in brown anole population in the presence versus absence of the curly tails. Now, the brown anoles are no dummies. Immediately, they move up into the vegetation. I forgot to tell you, curly tails are almost exclusively terrestrial. So the brown anoles are getting out of their way. I think it's interesting that the perch height kept on increasing throughout the experiment. We don't know why it kept on getting higher, but it did. Unfortunately, Hurricane Floyd, our study site is right here, wiped the experiment out, washed the islands clear. The brown anole populations survived, but we had to wait 5 years for the islands and the brown anole populations to recover. The curly tails were all wiped out. But we were finally able to do the experiment a second time. This time, knowing what would happen, we modified what we did in one important way. Again, we had 12 islands, 6 of them, the same 12 islands, 6 of them randomly assigned with the predators. But this time we caught all the brown anoles, or at least many of them on each island, measured how long their legs were and other traits, and gave each lizard an individual mark, the equivalent of a bird band. What you can see here is we're injecting just under the skin with a non-toxic rubbery surface. And so each lizard had a different combination of legs in which there was something and the color. This one has lime green in the left forearm and orangey red in the femur on the right side. And so each lizard had its own marker. And so what we could do is we could come back later and see who had survived and who hadn't, and whether survival was a function of limb length. In other words, test whether natural selection had occurred and whether natural selection differed between islands with and without curly tail lizards. So let's go back to Howard Hughes, if I can get this to work, and see what they have to say. Oh, I hit the wrong one.
Jonathan wondered if he could
And now it's
also
still
create
running.
an experiment
It's a disaster.
that would lead natural selection
All right, I got to go
favor
back
longer
there
legs.
and shut it up.
He found islands that
All right.
already had anoles that lived
I'm in the
primarily
wrong
on
place.
ground.
This is bad. Sorry. This is the worst
His team
job
caught
I've done
and
on
measured
this.
each lizard.
Oh,
Then he brought in a predator.
my apologies. I will get
A
this straight in a
species of larger
second.
curly-tailed lizards that eat
Ah.
anoles.
This is really
Here's the bad
embarrassing. All right. Got rid of that. Back to the share screen on the Zoom. All right, here we go.
After 6 months, he returned and discovered that on average, the survivors did have longer legs. They were better able to elude their foe. But after 12 months, Jonathan's team returned again and discovered that now most of the lizards were living high in the bushes to escape the predators. And on average, the survivors had shorter legs
Generation after generation, we're going to be up in the bushes. The shorter-legged lizards will do better, and we expect the population to evolve shorter legs.
Jonathan had seen how predation can create strong selective pressures for longer legs on the ground and shorter legs in the bushes.
Okay, so just to recap what that showed. The ecological effects were the same. In the presence of the curly-tailed lizard, the anoles got, they did not stay on they came up into the trees, off the ground. We measured it in a different way. You can see the difference right here. And we measured something called a selection gradient, which basically it measures the difference in the trait between the survivors and those that didn't survive. And so if selection favored longer limbs, then if the survivors had longer legs, you would have a very positive selection gradient. If the survivors had shorter legs than the non-survivors, the selection gradient would be very negative. Whereas if there was no difference between survivors and non-survivors, the value would be 0. And here's what we found. In the first 6 months, in the presence of curly-tailed lizards, selection was strongly favoring hind limbs, whereas in the absence, the control islands, there was no selection. So we had shown that selection differed, that the curly-tails were imposing selection for long legs. However, our prediction, we know that the lizards are moving up into the anoles are moving up into the trees, and as they move higher, they will be using narrower surfaces. Here's in the presence of curly-tails from a previous experiment. We know how anoles adapt to narrow surfaces. They evolve short legs. So our prediction was, given enough time, selection would flip, and in the presence of curly-tails, selection would favor shorter legs. Well, to our surprise, that flip happened much faster than we anticipated. In the second 6-month period, now selection is favoring shorter legs in the presence of curly-tailed lizards. So this was a very exciting result, a reversal in selection over a 12-month period. Now, going forward from here, our expectation, it's not going to oscillate because the brown anoles are stuck up in the bushes. Our prediction is selection now will continue to favor short legs generation after generation. And we were very excited to see whether, in fact, the brown anoles would turn into twig anoles. They're using very narrow surfaces. Would they evolve the very short limbs of species that have adapted over millions of years to using narrow surfaces? Well, you're probably thinking you know what happened next. You're thinking there was another hurricane. You're wrong. There were 2 hurricanes. First, Hurricane Francis, September 3rd, 2004. 3 weeks later, a second hurricane, Jean, did a loop-de-loop in order to go over our islands. So the poor islands, the poor people that were hit by 2 hurricanes within 3 weeks, wiping out our experiment. Again, we had to wait 5 years to start it again. We did start the experiment in 2009. The poor lizards on those islands have been hit by 5 hurricanes in the last 12 years. The ones you may have heard of, the most recently Dorian, which was a disaster. The populations are hanging on, but we can't give you a further update on the curly tail effect. It's been so messed up by the hurricanes. Maybe if we get a few hurricane-free years, we may still find out, but I have no further updates on that at this point. So instead, I want to briefly tell you about a different study I did on the topic of founder effects. I'm not going to go through the idea of founder effects. Ernst Mayr is famous for it. The idea is that populations founded by a very small number of individuals may evolve in interesting ways. And I'd always been interested, as far as I'm aware, there had never been a study of founder effects in natural populations. And these anoles usually colonize islands by a single gravid female. So many of these populations probably get started by founder effects. What actually happens when that occurs? And so these hurricanes had cleared off a lot of small islands that we knew could support brown anoles. And so we decided to take advantage of the opportunity and to create 7 founder effect islands. Now, I know I'm supposed to tell you that we were testing all kinds of hypotheses, but the real truth is we just wanted to see what would happen. And so what we did is, I'm going to skip this, bunch of questions, we'll get to them there. We found 7 very small islands, such as this one. We took lizards from a large island in the background that occurred in a forest, and we just took a male and a female and put them on 7 islands. So here's some of our small islands to give you a sense. We're talking really tiny islands. And so the question is, what would happen and so there's a question, are there going to be random founder effect type things happening because you're starting a population with 2 individuals? But at the same time, the vegetation here is very scrubby compared to the much thicker vegetation on the forest they came from. So our prediction based on other work is that they will evolve short legs. And so would the founder effect get in the way of that, or would they still be able to adapt? Well, first thing, remember, we start with 2 lizards. The populations thrive. This we did not predict. We thought some of the populations would just wink out for demographic reasons, but you can see the populations did very well. We measured genetic variation based on microsatellite loci. This is a little bit of a complicated slide, but this square is in a principal coordinate space, the average of all the lizards that we sampled from the source population. And then we randomly took 2 individuals to put on each island. Those islands are represented by different colors. And you can see the red ones are both to the left of the mean, the purple ones to the above the mean, and so on. Here is the genetic founder effect just based on using 2 individuals, just random sampling. And what we found is that those founder effects persisted for several years. Here's the 2nd year, it hasn't gone very far. Now we changed the axes, but you can see most of the islands, you create this founder effect genetically and it persists over the course of several islands. So there was a genetic founder effect. Our means of measuring lizards had improved. We now have a portable X-ray machine, my favorite thing in the whole entire world. What you can see here is there's a lizard on this platform that has the digital receiver for the X-rays. Here's the X-ray machine. And we get these wonderful images. You see all kinds of cool things. Who knew that brown anoles eat lots of snails, but many of them had little snails in their bellies? Here's a female. You can see how big the egg is compared to her body size. And look at this guy. It may take a moment to figure out, but this is a baby lizard head. There's its spine. We found a number of cases of cannibalism of these anoles. In another place, we found a whole bunch of lizards that had their legs chopped off, their forearms. We don't know why. We think it was either crabs or seabirds, but just one island was the island of the one-armed bandits, I guess. So all kinds of cool things with these X-rays. This is Jason Colby. We also looked at their toe pads with a scanner. And I'm not going to talk about their dewlaps, but Manuel Leal had a portable spectrophotometer to measure dewlap color. Well, there was also a founder effect in hind limb length. This is the 2 lizards and the F1 generation showing the difference in hind limb length 1 year after we set up the experiment. So you could see pretty substantial differences. So our prediction was, despite this founder effect, that the lizards would get shorter legs. And that's exactly what we see. Across 3 years, all 7 populations show a decrease in limb length. However, interestingly, not only is there a year effect showing they're getting shorter legs, but there's also an island effect. So this is the founder effect from the start of the experiment, and their limb length in 2006 predicts statistically their limb length in 2009. So the founder effect was still evident even as the species were adapting. So that was the interesting finding, that founder effects do occur, but they don't preclude adaptive evolution. Of course, our prediction would have been, probably given enough time, the founder effect, the evidence of it would be washed away by selection and evolution. I should show, here's the difference compared to our plasticity experiment showing the magnitude of the change. Once again, hurricanes finished this one off. Here's Hurricane Irene. Here's Hurricane Sandy, which wiped out 5 of the populations, ending the experiment. So briefly, I want to very quickly talk about 2 more recent studies that have gone on in the lab. Both of them have been done by postdocs in the lab. Both of these studies were experiments that I begged the postdocs not to do. I said they're terrible ideas, they won't work. Both of them were both hard-headed, pig-headed postdocs. They insisted on doing it, and turns out they were absolutely right. So I think there's a lesson in there, but I don't want to explore what that is. The first study was by Oriol Lapiedra, a fabulous scientist, now back in his native Spain, who was interested in, is there any selection on behavior on these islands? And so basically, he reasoned that lizards probably differ in their behavioral tendencies, what sometimes we call personality. There might be lizards that are very willing to do bold, risky things, and others that are more cautious. It's always dangerous putting human labels on these things, but that's the idea. And so imagine a lizard on an island. How willing is it to run out away from safety to forage for food? And some will be more willing to do that than others. And so you might imagine that there is a normal distribution of behavioral tendencies, and that in the absence of predators, the lizards that are more willing to run out there right away will do better because they'll get more food. But in the presence of curly-tailed lizards or a predator, those ones might get eaten and the more cautious ones might survive. That was the genesis of his experiment, to ask whether there are behavioral differences and would curly tails exert a selective force. So to do that, we went to the Abaco near our study site. He caught a bunch of lizards. He developed an assay to measure their propensity to forage, and then put the lizards on the island. Here is the assay. You catch a lizard, you put it in a box. This brown thing is a door. And the box this is in a little arena with a little tree. You then open the door, but put a curly-tailed lizard in a bag right in front of it. So it opens the door and says, oh my God, there's a curly-tailed lizard there. Then you remove the curly tail and you basically see how long it takes the lizard to come out and how long he sits out in the open before running up the tree to safety. That was his assay. It turns out it's a pretty repeatable assay when you run a lizard several times. It is a repeatable behavioral trait. So he caught 274 lizards, measured the behavior on each one, and then randomly assigned them to one of 8 islands that we created populations on. 4 of those islands subsequently got predators, the other 4 didn't. And so the question is there any selection going on these behaviors? Well, I should say the islands, the same sort of things we found elsewhere, that in the presence of predators, there was lower survival. And in the presence of predators, the lizard shifted higher in the trees, just as we would expect. So let's focus on the trait of how long they came out of their box after they'd seen a predator. The predator's removed, but how long do they stay on the ground before going up in the tree? And so remember, all these lizards, that has been measured in the lab, and then you put them in the field and you just ask who survives and who doesn't. And so it turns out that on the islands without curly-tailed lizards, there is no selection on propensity to stay out in the open. However, on the islands with curly-tailed lizards, there is very strong selection. Those lizards that stay out in the open for a long period of time do not survive very well, presumably because they're staying out in the open and getting eaten by the curly-tailed lizards. So this does show that selection does operate on behavior in these lizards. Now, of course, we would like to know, does that lead to an evolutionary change in behavior over enough generations? Another hurricane, Hurricane Dorian, but the good news is the lizard populations have survived. And so Oriel will be going back there soon to see if there is any evidence of actual behavioral evolution driven by these predators. Finally, one last quick study done by Colin Donahue, who was a postdoc in my lab. He's now moved on to a postdoc at Brown University, and Anthony Harel from the Natural History Museum in France. And this study took place in the Turks and the Caicos to the southwest of the Bahamas and focused on 2 tiny islands there, Water Key and Pine Key. The focal lizard was a species convergent with Anoles sagri, the brown anole, very similar looking, but it's a different species, Anoles scriptus. Now, these islands were overrun by invasive species, by rats, cats, and goats. And the conservation group, Fauna and Flora International, was embarking on a program to remove the invasive species. We thought this was a great opportunity to see how the lizards would adapt as their ecosystem was restored, as the vegetation changed, and so on. And so to get baseline data, Colin and Anthony and a couple others went down there in the late summer of 2017 and just measured the standard stuff we measured on anoles. This is the baseline data. They left on September 4th, 2017. 4 days later, Hurricane Irma went directly over these islands. And 2 weeks later, Hurricane Maria also went directly over these islands. So Colin and Anthony had pretty unusual data. They had censused these populations right before the hurricane. They went back out to see whether the population, whether there's any change in the morphology of the populations as a result of the hurricanes. And so they packed their bags and went out there. To cut a long story short, the surviving lizards had bigger toe pads on both islands. And you can see in the forelegs and the hind legs and on both islands, suggesting that the hurricane had been an agent of selection, that lizards that could hang on better had survived, whereas the ones that couldn't hang on as well had been blown off the island. And the fact that it occurred on 2 islands made that seem like a reasonable possibility. Well, I can talk more about, there are other possible explanations, none of which seems compelling, but let's assume for the moment that selection, hurricane-driven selection, actually might lead to larger towpaths. It has long been known that mainland anoles have smaller towpaths than island anoles. And people always ask me, well, could this be because of hurricanes? And my answer was, nah, hurricanes don't come along that often, and it seems unlikely that you could sort of a macro evolutionary result of the occasional hurricane. Well, Colin wanted to follow up on this. I thought he was wasting his time. And he did this in two ways. First, for 13 populations of the brown anole throughout the Caribbean, he was able to get an index of hurricane activity, how often hurricanes had hit different places, thanks to the work of a meteorologist who was a co-author on our paper. And the color red and the intensity is a measure of hurricane intensity. And then he went and measured towpad size. And sure enough, wouldn't you find that populations on islands that had been hit harder by hurricanes had larger towpads across 13 populations of brown anoles. Then he did the same thing throughout the entire anole radiation in the Caribbean and Central America. And he gets the same result. Even if you exclude the mainland anoles and just look in the Caribbean, species on islands with hit more by hurricanes have bigger towpads. So this may well indicate that hurricanes could be I'm still not entirely convinced, but it does suggest, at least possibly, that hurricanes are a selective force driving the evolution of longer towpads within and even between species. Well, to sum up, I never thought my career I'd be spending so much time talking about hurricanes. Certainly that was an unintentional thing, but we now know a thing or two about hurricanes. It's certainly been very fascinating. I am hoping that the hurricanes will stop, both because the poor people who live down there have been so pummeled by them, but also because we would like that experiment with the curly-tailed lizards to actually run long enough to see what happens. But at that, I think I will just wrap up and say thank you very much for this invitation. I look forward to speaking with some of you tomorrow, and I'm certainly happy to take questions.
That was great. Thank you so much, Jonathan.
Thank you.
So if you would like to ask questions, if you don't mind just using the raise your hand button or put it in the chat, and we can go from there.
Jennifer, do you want to moderate or should I do that?
What would you prefer?
Why don't you take charge?
Sounds good. Will do. I have a question to kick things. Oh, good, Leroy. Yeah.
Hi, Jonathan. My name is Leroy. It's nice to see you, and I've been an admirer of your work for a long time. I work in streams and rivers, so these systems are sort of isolated at the whole basin scale, but within a particular river system, there's a lot of continuity in gene flow. And I'm wondering about your results. They're so striking. And one of the things that strikes me as a river ecologist is how isolated your populations are. And of course, you have very strong selective pressures like predation going on. But I wonder how you would, and this is not really a well-formed question, but it's more of just how you think about this, how you think about your results in the context of more mainland systems where there's a lot more gene flow and maybe, rescue after disturbances or whatever. And of course, I'm just now rereading from a long time ago, David Quammen's book about the Song of the Dodo. And that seems to be his sort of main theme, is sort of fragmentation of the planet and how that's sort of making islands out of terrestrial systems as well. But just wonder how you think about, in terms of rapid evolution to strong selection, how that occurs on mainlands versus versus your islands.
Well, I think that's a fascinating question. And certainly in a situation where you have strong and contrasting selection, we do know that sometimes gene flow can be strong enough to keep populations from becoming too differentiated. So it's certainly a possibility. One of the things that we have failed to do is to get a good sense on these islands about how much gene flow there is. Our assumption was that, these are islands. Yeah, the lizards get out there occasionally, but there can't be that much gene flow. But that one hurricane, the first one where we were there, it was really astonishing how much stuff was in the water the next day when we got back out there. The water was just full of branches and mats of debris and so on. We didn't find any lizards on that, but you can well imagine them floating around. In fact, we did find spiders that we had never seen before on these islands that were only found on the big island that showed up immediately. So they were getting blown around. So one can well imagine that storms could be not only destroying populations, but serving as an agent of dispersal. Lizards, incidentally, can float and they can swim a little bit. So it's certainly possible to imagine. We did a microsatellite study a few years ago, which indicated surprisingly high levels of gene flow. And I'm not sure I entirely believe it, but if those results were correct, it suggests that there is a fair amount of exchange. Some of these islands, I should point out, are pretty close together. I mean, we're talking 100 meters. So one could easily imagine if a lizard falls in the water, it could end up at the next one. But the reason we haven't done more, and this is probably our lack of ambition, is that, of course, there's so many islands there that you'd have to, it would be a really big job to really sample enough of the islands to have probably an accurate idea. But I guess it's a long way of saying we don't know, but I think gene flow really is an important factor that needs to be considered. And even in our own study where they do seem isolated, they may not be as isolated as we seem.
Thank you.
If I can tell you just one story, it's not even my own work, but I love this story. The island of Anguilla, which is to the south of the Bahamas, to the south of Puerto Rico, historically never had green iguanas. And one year in 1996, periodically every year in the hurricane season, stuff would wash ashore on Anguilla and other places. Well, at one point there was this big mass of vegetation washed ashore, but it had 15 iguanas on it. And the local people saw it wash ashore, saw the iguanas get off, and now there are iguanas on Anguilla. They've established themselves. And the question, well, where did they come from? Well, this was the first of the busy hurricane years in 1995. So could in theory have come from anywhere. But in this mass of vegetation, there was a street sign, and the street sign was in French. And so there were only two islands it could be, Martinique or Guadeloupe. And there'd just been a hurricane that had gone over both of those islands. And it turns out it was probably Guadeloupe. So, I mean, these things do occur. And so if it can happen with green iguanas, certainly brown anoles can, over much smaller distances, can travel as well.
Thank you.
You're welcome.
So is that the first study to actually use street signs instead of like genetic markers or mark recapture to determine dispersal?
The amazing thing is that they did not put in the street sign thing in the paper.
Oh, that's a missed opportunity.
And I asked the lead author, why not? And she just thought it didn't sound scientific enough. And I thought it was the best part of the story.
Cameron.
Yeah, Cameron, go for it.
Hey, Jonathan, how's it going?
Thanks for staying up late.
No problem. Great to just get the full overview of your amazing career, and yeah, huge fan. I guess one question I have for you, kind of following up maybe a little bit on what Leroy was asking, is so you have this kind of dynamic system where populations are kind of blinking out and then getting recolonized, presumably, between islands. And then at the same time, you have these very small founding populations that presumably get established and then become locally adapted to the different substrates in terms of limb morphology, behavior, everything else. And it strikes me sort of in the same way that guppy populations have become established from very small founding populations and then shown repeated parallel adaptation to their environments. it seems like these results in some way kind of defy the fundamental tenets of population genetics, that these very small founding populations with very low genetic diversity can still have this capacity for not just for evolving at all, but then also to show, repeated predictable changes in response to the environment. And I'm just curious what if you've given that much thought, like I know you think a lot about the determinism of evolutionary patterns and how you reconcile the sort of small effective population sizes with the sort of deterministic outcomes.
Well, I think that's a great question. It's troubled me the entire time that we've been doing this research. Now, one thing I will point out is that these are quantitative traits we're dealing with, so that the limb length is affected by many different genes, loci. So perhaps that it's less likely to have no variation. But still, classic theory suggests that there would not be genetic variation for these things, and that I think is surprising. And but there are many other studies that have found similar sorts of results. And I will say two different things about this. One is that I'm just going to have a what's the word? A look to authority. I asked Dolph Schluter this question 2 years ago. He said, "Does this trouble you at all?" He said, "No, it doesn't trouble me. There's enough genetic variation. Selection's powerful enough. So if it's okay with Dolph, it's okay by me." But I guess the question is, what is the alternative? And the alternative is that it's not genetic change. One question that I do get a lot, particularly when I talk to public audiences on this work, is what about epigenetics? And that's the I certainly don't want to go down that road, but a lot of this evolution is occurring very rapidly. I just wonder whether we might find that perhaps there's something there. I even saying that, I didn't say that, don't quote me on that. But I guess the bottom line is it does seem awfully fast. And would be really great to actually try to find some of the genes underlying limb length, because then we can directly get a sense of what's going on. But I lose sleep over this sometimes, because it is a question. How does this occur so rapidly with presumably such little variation? Oh, I do need to tell you one other thing though. For what it's worth, when you put one male brown anole and one female brown anole on an island in May, you're probably going to have several males potentially contributing to that population, because brown anole females mate multiple times, they store sperm. And so the founding populations aren't quite as small, but still they're pretty small. So I've kind of rambled all over without giving you much of an answer, but it is a surprising result, I would agree.
These also store sperm, so you could have similar effects where a single female might have a lot more genetic diversity than you would expect otherwise.
Yeah, that makes sense.
As a quick follow-up to Cam's question there too, can you remind me, so did the founders come from the same location?
Yes.
So they're more likely to be more genetically similar anyway. And then potentially, right? Yeah. And then in terms of, not knowing yet the genetics behind limb length, I mean, just the possibility of having, multiple genes and other, continuous variation contributors, depending on how things are mixed up from generation to generation, I could potentially see how that could then lead to particular combinations over time, right?
Yeah. One of the things that I have tried to do over the years is to find some good traits that we could monitor for drift type processes as a foil. And we never found a really good one, but there I can say qualitatively, some of the lizards have little red caps on their head, little orange caps. And some of our populations, all the lizards have orange caps, and other ones, none of them do. And I am presuming that that is just a trait that's been experienced a founder effect and has led to differences. I wish we had a trait that we could monitor better, because it would be nice to have a range of different ones.
That would be interesting. Ruth?
Almost did the forget to turn off your mute thing. Great talk. Such a fascinating set of work. So I'm going to follow on from Leroy and Cameron. First, like, I'm not so surprised that rapid evolution happens from small population sizes, whether it's 2 or like 4, depending on the multiple mating, 6 individuals. Like, that's, if the population grows rapidly, most of the genetic variation is retained. And one thing that I think that I wonder about with my own work is how much a simple environment drives rapid responses to selection. And so going back to what Leroy was asking with respect to terrestrial, like large land masses relative to islands, we are getting fragmented habitats, but the environments are, like, the communities are potentially more speciose, and there might be more conflicting kind of selection pressures that populations are experiencing. And so I was just wondering if you could comment on that in terms of the generality of how rapid evolution happens and how important it is for the ecological phenomenon we see.
Well, I've never thought of it in that way, but I think you're absolutely right. I mean, that is the reason to work on these simple island systems, because there are only a few actors and you can really isolate those phenomena. But I think you're right that for that reason, the selection is probably much more unidimensional rather than being hit, from many different sides. And it seems very reasonable what you're saying, that the stronger and more unidimensional selection is, the more likely you are to get a rapid response. So I think you're on this. I would agree with you. And it is these islands, you saw the pictures. There's just not a lot of different things happening to them there. The thought of, I'd love to do an experiment like this in Central America, but there are so many different things impinging on the lizard, and their populations are smaller. And they're also, they're scaredy cats. You can't find them because they're so afraid of all the predators. So yes, I think you're absolutely right. That's a very interesting perspective.
Yeah, it's fascinating work, and I'm envious of those islands. They're super cool. They're simple yet real systems.
Yes, exactly. And I didn't show you any of the ecosystem stuff that Dave Spiller and Tom Shaner have been in charge of. But yes, they're real systems. They're all there are, there's a food web there with interesting interactions and so on. But yet you can really get a handle on it. I do have to give full credit to Tom Shaner as the one who recognized the opportunity that these islands presented, and Dave Spiller, who was certainly from the experimental work with him from the very beginning.
Jonathan, I had a question. Can you hear me okay?
Yep, I can hear you.
Okay, so when you were talking about small isolated populations, my thoughts kind of went to Sewell Wright's ideas. And, he would say that those small populations would give an opportunity for the different populations to stumble on different trait combinations that might be equally good solutions to the problems the ecological problems on those islands. And so I was wondering, did you ever see any evidence in these kind of replicated evolutionary experiments that different lizard populations were kind of stumbling on different epistatic trait combinations?
Well, actually, one of the things we were most excited about with the founder effect was what effect it would have on the dewlap, because the dewlap is the color of the dewlap and pattern is used in species recognition. And so one could imagine that if you had founder effects that caused the dewlaps to be very different in color just due to some random whatever, I mean, that's not just differentiation, that's potentially speciation. And so that was actually the real driving force behind that experiment. And there was a little bit of differentiation. There's a little bit of a founder effect initially, but it went away among the 7 populations. And so, I mean, the answer is no. That was what we were kind of interested in looking at, and we didn't see any sign of it. I sometimes wonder whether if we measured the heck out of these lizards and just kind of measured every sort of thing, every trait, and now you get their genomes and look at it at that level, whether we could learn a lot more than we have from the things that we have measured, and maybe you could. But anyway, a long way of saying we did not see evidence of that. We were kind of hoping the dewlap might show something like that. I was hoping to get create a new species out in the Bahamas with a yellow dewlap or something. No new species, sadly.
Okay, thanks.
Thank you.
Yet.
Yet.
Well, it's interesting too, thinking about some of the behavioral characteristics, behavioral traits that could be responding to this as well. I mean, those X-ray images you showed of having snails in their diet and even, cannibalizing juveniles. How does that compare to what you know about their diet on some of the larger islands or mainlands? And granted, I'm going beyond brown anoles, but just kind of anoles in general.
Well, let me answer that in a couple of ways. Number one is anoles are known to be cannibals. That wasn't a surprise. There's lots of reports of different species. Basically, an anole will eat anything small enough to swallow. And so that's including their own young. So it doesn't particularly surprise me. What does surprise me, so I wrote a book on anoles, as you probably know, it was this thick. It had 1,400 references. Anoles have been really very well studied. And when I set out to write that book, one of the things I thought I would do is, look, I'm going to read all these papers and I'm going to just synthesize all the work on diet and kind of understand variation in diet and come up with some interesting synthetic findings. And what I came to realize is, even though these lizards have been studied intensively for 50 years, many basic aspects of their natural history still are unknown. And diet is one of them. Yes, there have been a bunch of studies, but it's hard to study one species where one population has been studied in the mountains in the summer and another species has been studied on the coast in the fall. From all of this literature on diets, really all I could say is they eat insects and they tend to have pretty broad diets and they eat other things. There was no bigger revelations. And it might be that for the most part, they're generalists, but we really don't have enough data to really in any informed way talk about the nuances of how diet varies by season, by size, by humidity, by whatever. It just hasn't been studied. I mean, I would say, all right, there are 400 species of anoles. I would say that 10 of them have been studied pretty intensively. The brown anole, the green anole, a couple others. Maybe another 40 have been studied a little bit. And most of them we don't know anything about at all. And it's, and this is a group that's been intensively studied. So I got on my little soapbox there, but there's so much natural history just to be done before we can ask questions like that. And diet, how can we not know more about diet than we do?
Well, it's interesting too, thinking about it from the perspective of those small islands, right? Because thinking about what is available as a food source, and if we assume a generalist type of diet in the brown anoles, and then putting them on an island where they can take advantage of that, but then is there some sort of selective pressure going on there for those that will eat snails, for example, or do all of them eat it? Those kinds of questions too.
So one interesting thing that I'm not sure we've published or even conclusively demonstrated yet, but in the absence of curly tails, the lizards will be more willing to run around on the ground. And that includes going to the shoreline of these little islands where, at low tide, the little amphipods and stuff are exposed. And so that the predation will affect their willingness to eat different types of prey, or their ability to do it. And in turn, the islands are subsidized by all the seaweed that gets washed ashore that all the amphipods eat on, that is much more available when there are no predators around, or much more accessible. So there are certainly, what you're suggesting, the behavioral tendencies combined with the differences in the communities certainly has relevant impact.
That's fascinating. Are there any other questions?